It's my pleasure to talk about the company called Longboat Explorers. My name is Månsson. I am an equity partner and the CEO of the company. My background is that of a pharmaceutical executive. I used to head business development in Leo Pharma. That was my last job, and in that job obviously I was looking at these kinds of companies as potential acquisition candidates. So hopefully I will be able to use the experience that I have in order to turn Longboat Explorers into an acquisition candidate a few years down the road. So that's the overall objective from a financial perspective. So Longboat Explorers, what's in a name? Well, the connotation is obviously that of a Nordic company, so that's the Viking ship, the longboat. But we explore a different area. We also push frontiers, but we don't push frontiers in the geographic world. We do push frontiers in terms of the science, and the science is devoted to becoming the first commercial supplier and developer of amniotic fluid-derived mesenchymal stem cells and human pulmonary surfactant, and I will tell you all about that in a second. I should also say that the company is backed by Christopher Reyes, who's a serial entrepreneur. He's taken many companies to the successful listings on the Stockholm Stock Exchange. And he's also the chairman of the board. So stem cells, I think it's a very exciting area to be in. On the left of the screen here you can see just a cutout from a news site that I subscribe to, and I suppose you can't see the text there, but it's all about the fantastic things that you can do with stem cells. So on the top left corner there's a guy who's having his sight restored, blind people having their sight restored. Miraculous effects, and all about building organs in the lab. So it's perfectly Jesus kind of stuff, this. But if you're more down to earth as I am, perhaps the most important news is on the right side, and that is the fact that earlier this year, in March of 2018, the first ever approval of allogenic mesenchymal stem cell therapy was approved by the EMEA. And that was a product that was developed by biotech company TideGenix. It's based on adult MSCs, mesenchymal stem cells, and we will be exploring neonatal MSCs. But that company got acquired by Takeda for €500 million earlier this year. So that is a powerful precedent for our cause. So what can we actually do, and what is it that we aspire to produce? We aspire to produce a better, and the key word is neonatal quality mesenchymal stem cell therapy, and we do that by harvesting these cells from an abundant source, which is amniotic fluid. Mesenchymal stem cells are used in a variety of diseases. In terms of clinical development today, there are over 400 clinical programs being launched in the world today, and they cover wide ranges of therapeutic areas, all from MS to inflammatory bowel disease and to degenerative diseases like arthritis, for example. From the amniotic fluid, we're also able to derive human pulmonary surfactant. And I don't know how familiar you are with surfactant, but basically a baby, when it's in the womb, its lungs are exposed to a lipoprotein called surfactant, which makes the functional organ of the lungs mature. And if the gestation doesn't last all of the 9 months, if you're born prematurely, you've not been sufficiently exposed to surfactants. And that leads to the alveoli collapse that you can see on the right side. And that is obviously a very, very serious condition that can lead to death and also impair quality of life down the road if you manage to survive. So the rescue therapy today is that you derive surfactants from the lungs of pigs or cows, and that is administered to prematurely born infants today. The problem is obviously that the surfactant structure is not identical from species to species, so there's an impairment of the efficacy of that therapy. But the more powerful problem is that obviously if you take a large molecule from a different species and give it to another species, there is a substantial immunological counter-reaction, which means that you cannot treat adequately. So our idea is to give human surfactant to human babies, and our idea on the stem cell side is to provide neonatal quality stem cell. And I'll explain in the next slide why that is superior. Over on the left we have a very schematic description of our technology. We utilize amniotic fluid from planned C-sections. That's very important because it's important that parents have the ample time to think about whether they want to donate or not. Of course, if the fluid is not donated, it is simply spilt on the operating table, so it's a waste product if it's not utilized like this. In terms of practicalities, how this is done is just, instead of making the full incision, first there is a very small incision made and the tip of the device is inserted into the uterus and the amniotic fluid is siphoned from there. And then it goes into our airtight sort of ecosystem where we filter and centrifuge and are able to produce the two components from this fluid. And the good thing about using amniotic fluid from planned C-section is also that it is so abundant. Obviously, the other alternative is to make healthy volunteers donate tissue from which you can derive stem cells. But the commonness of planned C-section, of course, makes that an ideal situation because you have healthy people in a healthcare situation already. And the mesenchymal stem cell market that we're targeting is currently $1.5 billion US and growing very, very quickly. And on the other side, we have the surfactant market, which with the animal-derived products is about $300 million US. So this is a key slide to explain to you why neonatal quality stem cells are better stem cells. The potency of neonatal quality stem cells is much higher than the potency of adult quality stem cells. And here we've made a few experiments and we've compared to what is used today, which is basically adult stem cells, mostly from bone marrow and from adipose tissue. That's the red and the yellow line there. And the blue lines are neonatal stem cells, and the only superior quality stem cell that we have compared to is the fetal lung stem cell, which is coming from aborted fetus. So obviously that's not something that can be commercially available. And if you care to compare, for example, at the time point when we have 20 doublings, our competitors have about 10 doublings. And that difference is much more than perhaps meets the eye at first glance, because if you compare 20 doublings to 10 doublings, that's 1,000-fold difference. That's a huge factor. And obviously that is an indication of the therapeutic quality of our cells, but it is also an indication of the productivity that we can have using neonatal source stem cells. Another factor is that our cells are, to a large extent, able to display lung characteristics. And the reason for that is that most of the stem cells that you can find in amniotic fluid originate from the lung tissue. And why is that? Well, that's simply because the lung tissue has the largest surface area that connects to the amniotic fluid. The internal area of your lungs is actually the size of a tennis court, even though they are contained in your torso. So we believe that apart from our general advantage of having extremely proliferative cells, we have an advantage in repairing lung tissue specifically, which is what we will target in terms of our clinical development. So that's a lot of tech. Now let's talk about business. So what have we done and what do we aspire to do? Well, we have essentially two different business models, or relatively similar but different business models. Obviously, since these cells can be used in such a wide variety of different indications, we cannot cover all these indications in our own development program. So our idea is to license the use of our cells to other pharmaceutical companies for developing in areas outside of our own area of development. So if we choose to go for respiratory disease, we are free to license these cells to other companies who wish to develop them for inflammatory bowel disease, for MS or whatever. And that's the end goal, obviously. What have we done so far? So already before founding the company, there's a lot of conceptual development that has gone into this. We've done 2 bouts of R&D projects. One of those is published and the other one is sent in as an abstract and will be published later this year. We've done a lot of market research to have a really good set of potential customers for licensing. And we are now in the fall of 2018, and we're looking to refinance the company and to secure 2 to 4 million euros. And what is that needed for? Well, that's needed for upgrading and upscaling our production facilities. Currently, we have R&D grade facilities. We need to upgrade them to GMP facilities in order to be able to produce both for ourselves and also for our licensees. We need to do more targeted preclinical development to segue our way into the clinical development that we wish to undertake. And obviously we need to do business development in order to find our license partners. What we envision a year from now is, having climbed this staircase, that we will be soliciting another, probably larger amount to invest into the clinical program. So that's the funding that is envisioned currently. And as I said also initially, there is a good precedent for doing this. This is exactly what TiGenix did, but only for adult mesenchymal stem cells. We want to do the same thing, but for neonatal and superior quality MSCs. And they did this from 2011, when they started from scratch, basically not having any stem cell research prior to that. And they built a small platform, and they were able to be acquired for €500 million. Final slide, key aspects of the business proposition that I'm making here. On the left, this is a large and growing market. I'm talking about the mesenchymal stem cells now. It's about $1.5 billion and obviously climbing, and also anticipating a little bit further into the future, since there are so many clinical trials ongoing. If only a fraction of them actually make it to market as pharmaceuticals, the demand for having these cells will obviously increase. So being in the cell production business is arguably a good position. And if you look at the market, that's the pie chart. You can't see the labels, I can see, but the gray area there is bone marrow MSCs, which is an adult source. And the yellow area, which is increasing at the moment, is adipose tissue mesenchymal stem cells. And both of those are adult stem cells. So about 75% of the market is supplied by adult stem cells today. And I think this will change into neonatal stem cells in the near-term future. We do suggest that we have the right team over on the right side. As I mentioned, Christopher is very, very experienced in bringing companies to successful listings. We have our 3 inventors. To the left is Bjarne Woods, who is our Chief Scientific Officer. We have Marcus Larsson, who is a neonatologist and specializes in pulmonary surfactant. And we have Andreas Habst, who is the head of the Association for Swedish Obstetricians and Gynecologists, very influential in that area, and the one who has designed our device for collecting the amniotic fluid. So that, I think, is the final slide, and I'm open to take your questions.